bilayer with proteins that span the membrane, and they stick out on the surface. And those proteins on the surface are basically what we call antigens. Okay? And there are about 150 to 250 antigens present on the surface of your red blood cell. Each one of these proteins is coded by a specific gene, right? Because there are 250 different types of proteins on the surface. So far, we've been able to classify approximately 30 of these antigens. And you can follow them as they go from one generation to the next. Now, the ones that are most important medically speaking are your AB blood group, ABO, correct? And then the rhesus positive, rhesus negative, Rh plus, Rh minus, right? Those are the two that are medically important. But in addition to those important ones, there are others that are present. It's just we don't care about them medically speaking. But let's look at one of these groups that is not important medically, but gives you the idea of what is codominance. Okay, so ABO is one system of antigens on your blood surface. The Rh plus, Rh minus is another system. And then there is a blood group or blood antigen called MNN. Okay, the MN antigen. And there is a single gene that makes a protein, and this gene is called a gene L. Okay. This is not medically important, so we never study it with regards to blood transfusions. But there are two antigens, the M antigen and the N antigen. And there is a gene called the L gene, which codes for a protein that's present on the surface of the red blood cell. And there are two forms of the protein that are made by this gene, right? You can make a gene, L gene, that produces the M antigen, and a version of the L gene will produce a protein that makes the N antigen. So you have one or the other. So there are two alleles, the LM allele and the LN allele of the gene, two different alleles. So two allelic forms of the L gene, LM and LN, right? Okay, so if we were homozygous LM, what antigen would be present on the blood surface? The M antigen, correct? If you are homozygous LN, the antigen on the blood surface will be an N antigen. But what happens in the heterozygous condition? When you are LM, LN, the two alleles are together, what antigen would you expect to be present on the blood surface? If Mendel's rules are correct, it'll be either M or N, correct? One or the other in the heterozygous condition will be expressed on the surface. Well, in effect, what you find is that both are expressed. And that's why we call it codominant. Okay? So, with respect to the red blood cells, let's look at the relationship of genotype and phenotype. Okay? So your LM, LM, your homozygous LM, and on the surface of the blood cell, and then we're going to look at an LM, LN individual, and then the heterozygous LM, LN individual. The phenotype is the proteins on the surface of the blood cell. So if you're LM, LM, you're producing the M antigen on the surface of the blood. Those are these little black dots. If you're homozygous LN, LN, you're producing the N antigen on the surface of the blood. And those are these little triangles. So different form of this L gene. But then when you're heterozygous, you have both of them present. You have the M antigen as well as the N antigen. And because you have both of them being produced at the same time, you call this phenotype codominant phenotype. It's different from the incomplete dominance. The incomplete dominance is an intermediate trait between the two homozygous conditions. In the codominant, both traits are being expressed at the same time. So, slight difference in how you define it. Both proteins are being expressed in the heterozygous condition. Now, at the protein level, if you were to study most phenotypes at the protein level, not at the visual level of observation, they're almost always codominant. So the mutant enzyme and the wild type enzyme are both being made. If you study the presence of the enzyme, you will see that both are codominant. The wild type enzyme and the mutant enzyme are being made, basically. Most of the time. Of course, there are examples when you don't have that, but most of the time, at the protein level, phenotypes are always codominant. And that's what you're studying here. You're studying the presence of a protein. When you study the presence of a protein, you almost always end up with a phenotype that is codominant. Now, previously, question?
I was going to ask, they're both present, but basically one is just...
Well, at the level of the antigen, they're both present and they might both be functioning. They might have slightly different functions, but they'll still be functioning. Okay, so at the level of phenotypes, previously we were looking at things like flower color, hair length, or whatever. Those are visual morphological phenotypes, things you can observe with the naked eye. But when you go deeper into molecular phenotypes like this, how do you study these? You can't study these with the naked eye. There are tools we use to study molecular phenotypes. The blood group phenotypes are at a much finer level, and that's at the level of the cell. And so it's harder to observe. And so this is important. The phenotype is what you as a geneticist decide to study. You could say, I'm going to study the phenotype at the presence, the color of blood. Well, in that case, whether you are LM, LM, LN, LN, or LM, LN, you'll always be red, right? So it doesn't matter whether the gene is LM or LN, you're still going to be red at the level of observed phenotype by the naked eye. Antigens, we don't understand what. There are many antigens on the surface of many different cell types that we don't have a clue what their function is. These are just proteins, and it's usually not even proteins, but it's modifications on specific proteins. And these are glycosylations, basically carbohydrate moieties that are stuck onto proteins. The absolute function of most of these we don't have a clue for. Even the blood groups, ABO group. Okay, so how do we study these cellular phenotypes or molecular phenotypes? You use antibodies. So each of these proteins is slightly different. And so you can make an antibody that specifically recognizes either the LM version of the protein or the LN version of the protein. So you can have an antibody that recognizes one specific version or the other. And so you take blood in a little dish, in a little well, you put the red blood cells in those dishes, and you add the antibody that are specific. You'll have an antibody that's specific for the LM version, and then you'll add it to the little well. Or you'll add an antibody that's LN, and you'll add it over here. You'll ask if the antibody binds, and when the antibody binds, it'll change the color of the liquid in the water. So the presence of color gives you one phenotype, the absence of a color means it's missing. And so you use that. So in effect, you're back to a color, so you're back to looking at color, but you're using a probe that's a molecular probe in effect. So here is one example. This is the LM antigen, and this is the LN antigen. And you have a specific antibody that will bind the LM antigen. This antibody will not bind this version. It will only bind this one. And instead, a different antibody will bind this protein, but this antibody will not bind this protein. So if cells have this protein, this antibody will not bind, only this antibody will bind. If cells have this protein, this antibody will not bind, only this antibody will bind. And when cells have both proteins, both antibodies will bind. So, genotype, the antibody we are using, and the phenotype. Binding by the LN antibody, the LM antibody. And based on that, we're going to answer which antigen is present on the red blood cell. So, when you are LM, the antibody against the LN protein will not bind. The antibody against the LM protein will bind. And because of that, you will basically say that this cell has the LM antigen on its surface. Correct? If you are LN homozygous, this antibody will bind, the LN antibody will bind, the LM antibody will not bind, and you will conclude that this individual is producing red blood cells with the LN antigen. If you are heterozygous, LM-LN, you will basically have this LN antibody will bind, the LM antibody will also bind, and you will conclude that the phenotype of this individual is they have antigens that are producing LM and LN simultaneously. And so these will be codominant phenotypes. The heterozygous is expressing both proteins, and therefore with regards to the expression of the antigens, they are codominant. So now if you take a cross and you do a cross between individuals that are LM-LM to LN-LN, the F1 generation will produce individuals that are LM-LN simultaneously. So they'll be codominant. Now you can do a self-cross. You can take an individual that's LM-LN, cross to another individual that's LM-LN, and you will get individuals that are LM-LM, LM-LN, and LM-LN. And the ratio of this will also be 1:2:1. So it will look like the incomplete dominance ratio, basically. The only difference is that the phenotype is codominant, not incomplete. Questions? Yes?
So if you were to take a question about codominance, it would be something regarding the protein, so more on a molecular level.
That's correct.
If for the flower example, instead of red and no protein, it was producing a red protein and a yellow protein,
You would get, it would be a codominant phenotype. Absolutely. Yeah. Okay. So the phenotype ratios are the same as the genotype ratios. So they're like, in that sense, they follow the incomplete dominant rule. Codominant and incomplete dominance follow the same rules, basically. But keep in mind, the codominant phenotype is different from the incomplete dominant phenotype. There's a subtle difference, and it's different. Okay, so phenotype is what you study, what you decide to study. If you were to look at a fly with wings, and you were to just look at the wing shape, these would look similar. There would be no difference, correct? But if you were to focus with a little microscope at the presence or absence of this cross vein, you would say, here is a wild type with the cross vein, and here is the mutant that's cross veinless. It's a mutant Drosophila that's used all the time. And so you follow. But it depends on how you study it. Naked eye, you wouldn't see this. But with a magnifying glass or with a microscope, you would pick it up. And so phenotypes are what you decide to study. It's a very subjective definition. And that's what we're going into, basically. So you can have phenotypes that are morphological, things you can observe easily, or you can have phenotypes that are molecular or cellular that you have to use sophisticated equipment to study, including sequences. Okay? All right, we will skip this. It's just okay. So we're going to now. So this is where I want to take you. I want to make things gray for you before the midterm, right? So everything's been nice and clear, black and white. You know, it's always one or the other. Now I want to take you into the realm of real biology when nothing is as it seems to be or should be. Make it a little bit more tricky and difficult. But it's fun at the same time. And the classic example is sickle cell anemia in humans. We're going to use this example to show you how phenotypes are very subjective markers, basically, properties. Okay, it's a good example of the variation in dominance depending on how you study this trait. Okay, this is an inherited disorder caused by a mutation in a single gene. And that's in the beta globin gene. Okay, so hemoglobin is the constituent of the red blood cells that carries oxygen. There are two molecules, it's a heterotetramer. The two molecules of alpha globin, two molecules of beta globin. A single point mutation in the beta globin gene results in this phenotype. Okay. So HBA is the wild type allele, and you get a normal beta globin protein being made. And HBS is the sickle cell allele, the abnormal form of the beta globin protein being made. Fair enough? Okay. So now we're going to examine the phenotype of the two homozygotes, either big A, or one with the mutant sickle cell allele, little s, or a heterozygous. So homozygous wild type, homozygous sickle cell, and heterozygous. We're going to look at these three different individuals, but we're going to look at them in different ways. We're going to look at it at the level of the individual. We're going to look at this phenotype at the level of the cell. And then finally, we're going to look at this phenotype at the level of the protein. All right? Okay. Remember, you will determine something is dominant, recessive, incomplete dominant, codominant, whatever, in the heterozygous. Keep that in mind. And here we go. Okay. Hemoglobin homozygous, wild type, heterozygous, and homozygous recessive. Homozygous sickle cell. Okay. The first experiment we're going to do is the ability to carry oxygen at sea level. Imagine you're in Santa Cruz. You have three individuals. One of them is homozygous normal. One of them is homozygous sickle cell, and one of them is heterozygous. And we're going to measure the ability of these individuals, the blood of these individuals, to carry oxygen in Santa Cruz. Okay, and this individual is normal. This individual is normal. And this individual is anemic, unable to carry oxygen. So, based on this, what would you say? Is the sickle cell allele of hemoglobin dominant or recessive? Recessive or dominant? How many think it's dominant? How many think it's recessive? Right. It's recessive because in the heterozygous condition, the sickle cell allele is being masked, and the phenotype looks like the homozygous wild type. Correct? So, at sea level, this is a recessive phenotype. Now let's go to Colorado, up at 8,000 feet, and there's low oxygen levels, high altitude. Same three individuals. We haven't changed anything. We've just taken them to a different geographic location. We're going to ask the same question. Can they carry oxygen? This individual is normal. This individual is anemic. This individual is anemic. So now, what would you say? Is the sickle cell allele dominant or recessive? Dominant. Because now, under the heterozygous condition, you're getting the phenotype that's the same as the homozygous sickle cell. All you've changed is the location of your experiment. And the definition of what is dominant and what is recessive has changed. So you can see how subjective this is. It's all dependent on where you do the experiment and how you do the experiment that you will determine if something is dominant or recessive. Okay? So this is at the level of the individual. Now let's go to the level of the cell. Okay. We're going to look at the cell. Okay. What do the cells look like from individuals who are homozygous normal, homozygous sickle cell, and then heterozygous? So we're going to look at cell shape. Okay. So when you're homozygous normal, you have a normal shaped red blood cell that looks like this. That's my definition, diagram of a normal red blood cell. If you're homozygous recessive, or homozygous sickle cell, keep saying recessive, it should be homozygous sickle cell, you're going to be a sickle cell shaped red blood cell that looks like this. And then when you're heterozygous, you're kind of partially sickle. You're kind of sickle, but you're not. You're kind of circular, but you're not. You're kind of in between the two phenotypes. Okay? And so what you end up with at that point is that you are incompletely down. So at the level of the cell now, if you analyze it, the sickle cell allele is an incompletely dominant allele. Okay, so now you've looked at the individual, and at the individual it could be either dominant or recessive. At the cell level, it's incompletely dominant. So let's look at the protein level. Phenotype at the level of the protein. Okay. Okay. So what we're going to ask is the protein present or absent? So, if you're wild type, homozygous hemoglobin A, you have a single protein being made. This is a protein gel. There's a picture of a protein being made. That's the globin protein. If you're homozygous sickle cell, you have a protein being made that's slightly different. And it runs kind of different. Right? Heterozygous, both proteins. So now you would say you are codominant. So at the level of the protein, the heterozygous individual is codominant. So now you understand what I meant when I said that phenotypes are completely subjective, depending on how you do the experiment and where you do the experiment. That definition is only relevant for those conditions and no other. You can't say hemoglobin, that sickle cell is a codominant trait at the level of the cell or at the level of the individual. You can say that only for protein. So hemoglobin S and hemoglobin A codominant. And so, the way you classify this, at the level of the individual, you're either recessive or dominant. At the level of the cell, you're incomplete. At the level of the protein, you're codominant. Yes? Kind of gives you a perspective on how the word phenotype is a very vague term. Right? Not the genotype. The genotype is fixed, never changes. It's always hemoglobin A, hemoglobin A, or hemoglobin S, or hemoglobin A, hemoglobin S. Genotypes don't change, but the phenotypes do change. Okay. And then if you look at yet another phenotype of these individuals, their resistance to malaria. When you analyze this, you find that the heterozygous condition has a new phenotype that neither homozygous conditions have. So, malarial resistance, the normal hemoglobin individuals are sensitive to malaria. Hemoglobin S individuals usually die early because they cannot carry oxygen even at sea levels. But then the heterozygous individual is resistant to malaria. And this is actually a form of human evolution that has taken place over the last 10,000 years, basically. It's a classic example of individuals generating resistance to malaria when they live in a geographical location where there is a lot of malaria. There seems to be a preference for individuals who are heterozygous for the sickle cell allele because they can resist. They can carry oxygen most of the time. They're not as sick, and they're resistant to malarial infection, so they don't die from malaria. So, another example. So you can see how the phenotype, depending on what you're studying, the definition of the heterozygous completely changes from one to the other. And this is known as overdominance. You've got a phenotype that is even greater than either of the two homozygous phenotypes. Okay? So this is just a picture of the whole thing. It's called pleiotropy. That's the word. You don't have to remember this word. It means one gene influences more than one trait. Depends on what the trait is that you're studying. Oxygen carrying capacity, blood shape, presence or absence of a protein, resistance or susceptibility to malaria. All right, so we've got 15 minutes. I'm going to start you on something else. Now here, we're going to look at alleles that affect phenotype, but only affect the way it appears that they're being passed on from one generation to the next. So until now, we've been looking at phenotypes that are not lethal. Eye color, hair color, height of plants, the presence of wrinkles or no wrinkles on the seed, etc. Those are not mutations that kill something. But what if you had a mutation in a gene that would kill that individual? What happens then? Okay? So most of the mutations we've discussed do not affect the viability of an individual. So mutations for white eyes in Drosophila or wrinkled cotyledons in pea plants, they don't disrupt viability. The plants are fine, the flies are flying. Okay? And so a mutated gene is involved in determining eye color or seed shape or whatever, but it is not involved in a central process of viability. But let's imagine there is a mutation in an enzyme that's required for DNA replication. You can't replicate your DNA, you can't multiply your cells, you end up being dead. Right? So an example of this is a gene called RPN1. This is a protein that's involved in degradation of proteins, regulated degradation of proteins in cells, called the proteasome. So you can get a heterozygous plant that's RPN1 wild type, RPN1 mutant, cross it to a plant that's RPN1 wild type, RPN1 mutant, and you will produce seeds. 3/4 of the seeds will be RPN1 wild type with the mutant form, and 1/4 of them will be RPN1 mutant, homozygous. You take these seeds and you plant the seeds, right? What you will find is that 75% of the seeds will germinate and grow into plants. 25% of the seeds will not germinate and not grow into plants. And the 25% that are not able to grow, that are lethal, are this class here, RPN1 minus, RPN1 minus. So the homozygous RPN1 mutant is lethal, it's dead. The cells cannot grow, and so RPN1 is an essential enzyme. In the wild type state, as 2 copies or 1 copy, the cell survives, but in the absence of this, the cell is dead. Now you can study this very nicely in plants, because when you get the seeds, you can plant the seeds and you can see if the seeds will grow into plants or not. But what about animals? How do you study a lethal gene in animals? It's not like you can kind of look at the embryo as it's developing, right? It might die at the level of the 8 cell stage or the 20 cell stage or whatever. Now, how are you going to figure that out? How are you going to study it in the womb of the mother or the inside of a fly or wherever? Kind of tricky. difficult to do. So what are we going to do? Well, we're going to look at the ratio. And if you see a disruption of the normal segregation ratio, you can infer lethality. So, we look at a phenotype of Drosophila for a gene called curly wings. So normal wings in flies are flat, and in the mutant, you get wings that kind of upturn, kind of curve.